Good morning, children. Welcome to Class 10 Biology, lesson number 15. In this lesson, we will discuss the cell cycle and cell division. But before we go into studying the cell division, let us see the characteristic features of animal and plant cell. Animal cell. Animal cell does not have a cell wall, that is, the cell wall is absent. Centrosomes and centrioles are present. They are characteristic features of animal cell. Lysosomes are present. Plastids are absent. Vacuoles, if present, are small in size. And of course, the reserve food material in animal cell is glycogen. Now let us see the plant cell. Presence of a cell wall made up of cellulose is a characteristic feature of plant cell. Plastids are present and are of three types: chloroplast, chromoplast, and leucoplast. Vacuoles are centrally located and are usually large in size. Centrosomes are absent. Lysosomes are also absent in plant cells. Reserve food material is in plant cell is starch. With this in our mind, let us see what is cell division. Cell division is a process by which two or more daughter cells are formed from a single mother cell. Question arises, why do cells divide? Cells divide for growth. That is, when a cell fertilizes and becomes a zygote, then it grows to become a fetus and then a young child and an adult later on. Repair. Any type of damage to the body of an organism has to be repaired. It is also done with the help of cell division. Replacement. We know that RBCs have a lifetime of only 120 days, and after 120 days they are destroyed. They have to be replaced by new cells. And of course, reproduction. Cells divide to form the sperm and the ova, which lead to sexual reproduction. Now, types of cell division. There are three types of cell division. First, mitosis, which takes place in prokaryotic cells like the bacteria. In this, the chromosome number is unequal in the daughter cells. Actually, in this type of division, the nucleus divides into two daughter cells without the nuclear membrane disintegrating. Hence, both these daughter cells may get equal or may get unequal number of chromosomes. Next is mitosis. It is also known as equational division. In this, the chromosomal number in the daughter cells is same as that of the mother cell. It takes place for growth, repair, and replacement. Meiosis is also known as reduction division. In this, the number of chromosomes in the daughter cell is half that of the mother cell. It takes place in reproduction, that is, formation of sperm and ova. Cell cycle. Now, what is cell cycle? Every cell capable of division passes through a cycle which is called the cell cycle. The cell cycle is broadly divided into two parts. The interphase, the blue part that you can see in this figure, is the interphase. Interphase is the non-dividing preparatory phase in which the cell prepares for the next cell division. Interphase is further divided into three substages. G1 phase. It is the first growth phase. The cell grows in size. RNA and proteins are synthesized during this time. No change takes place in the DNA contents. That is, the DNA contents remains constant in this stage. After this stage, the cell goes into the next stage, which could be the G0 stage. That is, in this stage, it does not go into the division phase, or it goes into a resting period, or it may enter the next stage, which is the M phase or the mitotic phase. Now let us study the next part. If it does not go into the G0 stage, then what happens? It then passes on to the next stage, S phase. S phase. During this phase is also known as the synthesis phase. During this phase, a replication of DNA and synthesis of histone proteins takes place. Chromosomes are duplicated. Then comes the G2 phase. This is the second growth phase. In this phase, synthesis of RNA and protein continues for cell growth. Centrosomes replicate. Now the cell goes into the next phase, that is the M phase or the mitotic phase. Mitosis. The process of mitosis is studied under two parts, karyokinesis and cytokinesis. Karyokinesis, all changes in the nucleus that occur during cell division are collectively termed as karyokinesis. During karyokinesis, many events take place in a particular sequence. All these events are studied under the four stages: prophase, metaphase, anaphase, and telophase. Now let us study each one of them in detail. Prophase. In this stage, the chromosome becomes short and thickened and they become visible in the nucleus. Chromosomes have already duplicated to form two chromatids. The two sister chromatids remain attached to each other at the centromere. The centrosome splits. It can be seen in the diagram that they have split and they are moving apart. At the same time, the centrioles duplicate and the daughter centrioles together with asters start moving towards the opposite poles. Spindle fibers appear between the two centrioles. The nuclear membrane and the nucleolus start disappearing, and by the end of prophase, they disappear. The chromosomes get attached to the spindle fiber. This marks the end of prophase. As soon as the chromosomes attach to the spindle fibers, it marks the end of prophase. Then comes the next stage, that is the metaphase. You can see very clearly that the chromosomes have come in a straight line. The duplicated chromosomes arrange themselves along the equatorial plane. This plane is called the equatorial plane and also known as the equatorial plate or the metaphase plate. Next is anaphase. First, the centromere splits. In this stage, the centromere splits into two daughter centromeres. Thus, each chromatid has one daughter centromere. Now they are called sister chromatids or daughter chromosomes. The sister chromatids start moving towards the opposite poles. This movement is brought about by the contraction of the spindle fibers. The spindle fibers contract and pull the chromatids towards the two opposite poles. By the end of anaphase, the sister chromatids reach the opposite poles. As soon as they reach the opposite poles, it marks the end of anaphase. Next comes the telophase. Each chromatid now starts unwinding. Unwinding becomes thin and forms the chromatin fibers once again. The nuclear membrane reappears around each chromatin network, giving rise to two daughter nuclei. The nucleolus reappears in each of the daughter nucleus. The spindle fibers and the astral rays disappear. The centrioles duplicate and are surrounded by the centrosphere to produce the centrosome near each nucleus. A cleavage furrow appears in the animal cell. Next we come to is cytokinesis, that is division of the cytoplasm. As I have already said that in the telophase, the constriction appears in the cytoplasm, in the cell membrane of the cytoplasm. During telophase, a furrow appears in the cell membrane which deepens and finally splits the cytoplasm into two daughter cells. Thus, we have two daughter cells at the end of mitotic cell division. With this, we finish this topic on mitotic cell division. In the next class, we will discuss the mitotic cell division that takes place in plant cell and how it differs from that of an animal cell. Thank you, children. Take care of yourself and God bless you all.